So chapter 12, it's about eukaryotic organisms, including fungi, algae, Protozoa, and Helminths. So here we have a type of eukaryote, which is a fungus. This is Cryptococcus gatti. It is a fungus that has this shape, like a circular shape, and it is part of the phyla of Cryptococcus neoformans, which is an encapsulated fungus. So in the first part, we're going to define what are the characteristics of fungi. We're going to see the different forms of reproduction that fungi has, and we're going to see 4 medically important fungi, and then identify some harmful and beneficial effects of fungi. So we have over 100,000 species of fungi, and many of these cause disease in humans, animals, and plants. So, however, we have many species that are beneficial because it helps to recycle materials that decay, like, for instance, plants. And then we have a type of fungi that are the mycorrhizae, that they are located in the environment, like in rocks, and they form a symbiotic relationship with plants. So it helps the plants to absorb water and also nutrients while the fungi is protected. So fungi, it is known to be beneficial for us because it helps us in the production of food, for instance, bread. And then also we have the first antibiotic that it was purified and tried in humans as kind of a massive type of antibiotic was penicillin. Penicillin comes from a type of mold that is called Penicillium notatum. The study of fungi, it is mycology, and then fungi are chemoheterotrophs. So basically, they can obtain their source of energy out of proteins, out of carbohydrates, out of lipids. And then they are either aerobic or facultative anaerobes. If you remember, facultative anaerobes, they prefer oxygen, but they also can live in the absence of oxygen. If you compare the characteristics of fungi and bacteria, well, in terms of the membrane of fungi has estrals. And then estrals are absent in bacteria with the exception of mycoplasma. If you remember, mycoplasma is the only bacteria that doesn't have a cell wall, and that's why it's pleomorphic, so it can take many shapes. And then that's why they call them mycoplasma, myco for fungi. So now, in terms of cell wall, the cell wall of fungi has glucans, mannans, and chitin, but it doesn't have peptidoglycan. So these are carbohydrates that this cell wall of fungi has. And in terms of reproduction through spores, well, bacteria has endospores that are not used for reproduction. They are used only to create a new vegetative form when the bacteria runs out of nutrients, and then it releases the spores into the environment. It's a form of survival, if you want to see it that way. And then when it comes to spores, for fungi, in this case, they are used for sexual and asexual reproduction. And then metabolism. Well, in the case of fungi, it's limited, so it's heterotrophic only. It's not they are not chemoautotroph, for instance, for using CO2. And then they are either aerobes or facultative anaerobes. And then when it comes to bacteria, you have a lot of diversity. You have some that are heterotrophs or others that are autotrophs. Of course, we have aerobes, facultative anaerobes, and then we have what is called the obligate anaerobes. So the characteristics of the fungi, that it's not the spores, the vegetative form. So it forms two types of structures. One that it is molds and also fleshly fungi. So the fungal thallus or body has a collection of filaments that are long, and they have a specific shape that you can use them to recognize them when you look them microscopically. This will be called the hyphae. And all this collection of hyphae forms what is called the mycelium. And then some hyphae may have fragmentations internally, so these are called the septate hyphae, septate for wall. And then some of these hyphae don't have this segmentation, so these are choanocytic fungi. And then these bacteria, this fungi obtains the nutrients with aerial hyphae that is also involved in reproduction. So here we have a septate hyphae. So these will be the separations, and then you can see there in kind of a purple dot the nuclei. And then this is a choanocytic hyphae. And then you can have some, in the case of choanocytic hyphae, and as well, of course, in the segmented, you can have some ramifications of the hyphae. It depends so much on the type of fungi. And then when it starts to growing, this fungi, what it does, it just grabs, the spore is released into the environment, and then from the spore, you start protruding this hyphae, and then you form your adult form. So here we have a microscopic view of light microscopy of this fungi, which is Aspergillus niger. So this one, it has these tiny fragments in here, or tiny, filaments that are the hyphae, aerial hyphae. And then you have these spores. So if you grow it in an agar, which more than likely it is an agar that it is rich in carbohydrates, like saburodextrose agar, for instance, you can see two types of growth in the agar. So this is a top view of the agar. So all of these that you see here in whitish color, that it looks like if it was like cotton balls, like that little fuzziness. So this is the vegetative hyphae. And then these black structures that you see here will be the aerial hyphae. And it's called Aspergillus niger because that means black. And then another form of fungi, it is yeast. And then yeast are non-filamentous, so they don't have hyphae, and they are unicellular. Hyphae, it is fragmented or non-fragmented. So in this case, for yeast, we have budding yeast that divides unevenly, or fission yeast that divides evenly. So fission yeast, basically, when it divides, it gives you like the same size, while the other one, the budding yeast, initially it gives you like kind of tiny buds, but these tiny buds, once they get released from the other yeast, the mother yeast, then they start developing the regular size. And then we have some fungi that are dimorphic. So that means that you can have two shapes. So if you grow it at 37, it will give you a yeast-like form. But if you grow it at room temperature, 25 degrees Celsius, will be mold-like. Not all the fungi is like that, or fungi. Okay. I don't know how to say. Okay, so here we have a scanning electron micrograph of budding yeast. So here is the parent yeast, and then here is like a tiny bud, and then here is another bud. And every time that these buds are released and they form their own yeast, they will leave in the mother cell these bud scars. It's like the C-section. Scars. And then here we have fungi that it has dimorphism. So you have here the yeast-like form that forms at 37 degrees and a mold-like form that forms at 25 degrees Celsius. So life cycle. So they can reproduce sexually or asexually. So they can form spores then that detach from the parent cell as we saw before, and then they germinate into a new form. Now, if you have an asexual spore, this asexual spore will be reproducing by mitosis and cell division. So it is formed by hyphae of one organism, this type of asexual forms. And then we have within this type of hyphae, some hyphae that are not enclosed within a sac. These will be conidiospores. Other ones, they just fragment from the hyphae. And then these filaments, these hyphae that have these fragments, they have like kind of an articulation in which you have this hyphae that fragments from this long filament, this hyphae, and then you release them into the environment. So this will be what we call arthroconidia. And then if the hyphae, if the spore looks like a bud, this is called blastoconidia. And then chlamydoconidium, it's a spore that it is found within the segment of the hyphae. And then some of them, they look kind of very circular, and they're enclosed within a sac. These are called the sporangiospores. Like just, if you remember from that Myxococcus that has the sporangioles, so it looks similar to this. So it's like little spheres, but in this case, they're enclosed within a sac. So here we have different type of fungi with their different type of spores. These are all are sexual spores. Again, they reproduce by fission or by mitosis. So here we have Aspergillus niger. These are a scanning electron micrograph. And then at the end of this conidiospore, you have the conidia. This one, this basically, guys, is what you saw here before, but in scanning electron microscope. And then this light microscopy is kind of not very good at it, but it looks pretty nice even with light microscopy, this type of shape. So what does it remind you of? What vegetable? For me, it reminds me of that vegetable. I don't know about you, but. Broccoli. Mm-hmm. Yeah. And then in here we have Coracystis ulmi. So these are the hyphae. And then you have here the fragmentation, which are the arthroconidia. There are some other better examples than this. I don't know why they didn't show it. But it is nice when you see Let me draw it, because they didn't show it there. So in this fragmented hyphae, and there might be, of course, morphology genes that helps to form all these type of fragments, morphogenetic factors. But if this was a segmented one, and of course in each of them you have so you will have again some sort of articulation that makes this to move, and then one of these piece fragments, and then it goes out, and then it starts forming a new micelle or mycelium. And then here we have Candida albicans. So Candida albicans, as it's growing, as it's budding, it forms, of course, different sizes of buds, which eventually reach a particular size. But this budding, don't think it separates right away. And then it forms something that is called the pseudohyphae. This is very characteristic of Candida albicans. And there is some test that is specific for this to see when the yeast forms this type of pseudohyphae. And then these are blastoconidia. And then here we have Chlamydioconidia, which she has thick walls within this hyphae. And then this is also part of this Candida albicans. And then here we have this Rhizopus, which has at this end these large spheres and these tiny little spheres, which are the sporangiospores within this sporangiophore. Rhizopus causes a lot of allergies. And it has also this growth in the media that is similar to the vegetative form of Aspergillus niger. It forms like fuzzy colonies. And then when it grows sometimes in corn tortillas, guys. So when you separate the corn tortillas, you can, and then you have the growth in between the tortillas, you can see those long filaments, like if it were like a spider webs. Okay, so for sexual spores, they are basically having two mating strains that fuse their nuclei. And then you have, after this fusion, 3 phases of sexual reproduction. So basically you have something that's called plasmogamy, in which your haploid donor cell, nucleus, which is plus, it penetrates the cytoplasm of the recipient, which is the minus. So similar to the F plus, F minus. So that's the first step, the penetration of the plus over the minus. So this is plasmogamy. And then you have something that's called karyogamy, in which you will have the plus and the minus fusing to form a zygote. So in this case, you will bring whatever number of chromosomes the fungi has, mix it with the other nucleus, and then you form a single nucleus. And then it's similar to us, for instance, when you have the fertilization of an egg with a sperm cell. I mean, you have the male per nucleus and the female per nucleus, they fuse and they form a single nucleus, And in this case, you call it now a diploid cell because you will have the double of the chromosomes, and then the new structure is going to be a zygote. And then karyo, of course, stands for nucleus. And then meiosis, it will be when you have your diploid nucleus producing haploid nuclei to form more sexual spores. So that's the life cycle of reproduction with sexual spores. And now in terms of growth, so basically fungi, they live at very low pH, pH 5, so they are acidophiles. And then depending, of course, on the requirements and where they are located, they have to adapt. So they can be oxygenic or anoxygenic. For instance, molds, they are oxygenic, so they use aerobic metabolism. While most yeasts, they are facultative anaerobes. So they can live with or without oxygen. And then they like to grow in areas where you have high sugar content and salt concentration, so they are resistant to osmotic pressure. Their cell wall allows them to be so resistant for that. And then they can grow in areas with low moisture content. However, most of them, they prefer moisture. I mean, that explains why we end up with acid food, or sometimes candidiasis in the oral cavity, in the pharynx, in the vagina, because these are areas where we have a lot of moisture. And of course, at home, where do they tend to grow the most? In the shower, right? So it's an area of high moisture. And then when it comes to nitrogen requirements, fungi, they don't require as much nitrogen as bacteria, and they can metabolize complex carbohydrates, like starch, for instance. Okay, interesting question. Assume you isolated a single-celled organism that has a cell wall. How you determine that it's a fungus and it's not a bacteria? Excuse me?
They have a nucleus.
Yes, that's true. And what else? That was the easiest to kill question.
Peptidoglycan in the cell wall.
Yeah, the bacteria will have peptidoglycan, right? By the way, when you gram stain fungus, typically it's gram positive. They can stain with gram. That's too long. The next question. Let's move on. Okay, so medically important fungi. So we have different phyla. We have Mucoromycota, Microsporidia, Ascomycota, Basidiomycota. Mucormycota, it's what is called a conjugation fungi. It has concytic hyphae, so the hyphae that doesn't have the fragments, and it produces asexually an sporangospore and sexually a zygospore. And an example, it is Rhizopus stolonifer. This one forms black bread mold. So if you see green mold, of course, that's penicillium. If you see black mold, that will be Rhizopus. And that even grows in the shower caps. It's like that dark film that you see in the caulk. So here is the life cycle of Rhizopus. So you start here with the release of these sporangospores, and then that is going to germinate, form this mycelium, and then the mycelium can form more sporangospores asexually, and then it will have this aerial hyphae that produces this sporangium, that it has these little sporangospores. And then if it grows sexually, it will combine with some hyphae that it is minus, and then it will undergo plasmogamy, and then it will form these zygospores, and then it will form this karyogamy and meiosis, and then it will form another zygote, and then it will release the spores. Now for microsporidia. Microsporidia doesn't have sexual reproduction, but probably if it has a sexual reproduction, it occurs within the host. It doesn't have mitochondria, so it has to live inside a cell because it requires oxygen, and it's an obligate intracellular parasite. Something that it causes in humans is chronic diarrhea and keratoconjunctivitis, which is an infection in the eye. And then it can lead the person to blindness. So here we have the life cycle of microsporidia. So someone may inhale the spores. Those spores goes into the body, and then the body will make this hyphae these spores to have the right temperature, and then these spores is going to start forming a hyphae that is going to be endocytosed by the host cell, and once inside the host cell, you start growing these spores inside the cytoplasm of the cell. And then you will have the breaking up of these into small clusters of spores, and then you will release new spores. And then another person can inhale it, and another cycle can occur. So this is a transmission electromicrograph showing you what happens in these vacuoles, these little structures that are formed. So you will have some mature spores that form, and some other ones, they start forming inside these vacuoles. Ascomycota. It is a sac fungi. It has septate in their hyphae, and it produces sexually and asexually. So some of them, if they lose the property, because some of them they can lose the property to reproduce sexually, they become what is called anamorphous or anamorphic. So it is this type of fungi that when it reproduces asexually, it does so through conidiospores, and then when it produces sexually, it does so with ascospores. So in terms of the shape of the nucleus, well, it would vary based on if they have this structure that is like a sac that is called ascus, if it's different or if it's the same. So here we have the life cycle of Penicillium, which is one of these Ascomycetes. So we have this hyphae that has these extensions, and this extension will be the conidia, and then the conidia will be released, and then you will start germinating these to produce hyphae, and then these hyphae start growing, and then it can form these conidiospores, and then you can form more conidia. So that's the asexual reproduction. Now, in the sexual reproduction, if this mycelium encounters a cell that it doesn't have these genes that are plus, then it fuses with this in plasmogamy, and then you form a single nucleus in karyogamy, and then you form a structure that is called the ascus. So this, if you magnify this little part, of this cycle, you will have this seed-like structure that is called the ascospores. So these ascospores will be inside this ascus, and then you will have meiosis, then mitosis. You will produce more ascospores, and then your ascus is going to release the ascospores. So it's like a little sac that it forms during sexual reproduction. And then out of these ascospores, you form more hyphae. Basidiomycota, it's a club-type fungi. It has septate hyphae, and then it can produce sexually or asexually. It has conidiospores for asexual reproduction and basidiospores for sexual reproduction. They are formed in a pedestal that is called basidium. Base, it is kind of a name for a pedestal. So here we have the life cycle of any basidiomycete. This is asexual, and then this is sexual reproduction. So a fragment of the hyphae, once it breaks off from the vegetative form, the mycelium, will start producing new mycelium. And then it will elongate, forming vegetative mycelium when it encounters like a solid surface, and then it will continue growing and then forming more fragments and spreading to the surfaces that it has available. Now, for the sexual reproduction, again, if you have these fragments that has the plus, they will encounter some that they don't have this plus. During plasmogamy, the plus penetrates the minus, and then you will form a fruiting body, which is a macroscopic structure, a mushroom. So basidiomycetes are microscopic for the most part. This can be a type of fungi that it is called Amanita phalloides, for instance, that it is toxic for humans. It looks, it can be Amanita muscaria that looks like Amanita phalloides. And this Amanita muscaria, it is toxic for humans. The other type, phalloides, it looks similar, microscopically speaking. It has like a red cap with little speckles that looks like if you were snowflakes. And in the hippie era, people, they used to go to the field to have picnics, and then the hippies, they used to grab part of these Amanita phalloides because it had some hallucinogen chemicals in it. But sometimes you found in the field Amanita muscaria instead of Amanita phalloides. And this one has this chemical that can prevent the neurotransmission between neurons and muscle cells, and then you end up with depression of the respiratory system, and then, of course, your muscles won't contract. So people die out of eating all of these mushrooms in seeking of recreation. So then if you magnify part of this, part of the fungi, and you see it with the scanning electron micrograph, you can see this basidium, the base, that holds these little basidiospores. And then the basidiospores, of course, they start small, and they start forming these big structures. And then these ones can detach, and they form new hyphae. So that's the sexual life cycle. So fungal diseases, we have in general the name for fungal infections, which is called mycosis. We have 5 different types. The top one, it is the most serious, It is systemic mycosis. That means that a person will have the spreading of fungi within the body. This can go into the lungs, it can go into the brain tissue, and then fungi will grow in the brain tissue, form masses, and the person will have vomiting, headaches. It doesn't happen to everyone. It happens frequently to people who has immunosuppression. And the type of vomiting that happens because they have a mass like a tumor, but it's not a tumor, it's a fungi that it forms like a big ball inside the brain. It is not like the vomit that is kind of not so strong, it goes into your body or a short distance. It is what we call projectile vomiting. So that can spread feet away, and then the person can splash the surroundings with this type of vomiting. And then any subcutaneous mycosis typically starts as a cutaneous mycosis. So cutaneous will be a superficial mycosis, mycosis in the skin, an infection in the skin. And then that can go deep and then cause subcutaneous mycosis in the subcutaneous tissue, hypodermis, and then whatever tissue we have underneath. Well, it depends on what type of mycosis you have, but there are some subcutaneous mycosis that they don't go deep because the fungus doesn't have any enzymes to break down the skin. But we have some that they do. So they start as cutaneous, they can go subcutaneous. Now, some of them can go directly into subcutaneous because you have a wound. And if you have a fungi and even bacteria going directly into subcutaneous tissue, well, you will have that. Types of cutaneous mycosis can affect your hair, skin, and nails. So these collectively are called dermatomycosis. We have dermatophytes, species of fungi, like Trichophytum, for instance, that can live out of keratin. And then, if it happens frequently in the elderly, especially in the toenails, and then it can leave like the toenails very thick, and then as it's breaking down the keratin, it looks like it's fragmenting, and it does, it's actually, because, I mean, the fungi prevents the proteins to be close together as it's eating this protein. And then we have a specific mycosis, like the cutaneous mycosis that affects the hair shaft, like Trichophytum species as well can cause ringworm in humans and also in animals. It is very frequent to see young kids with patches of hair gone. And even not necessarily has to be the hair, but also can be the skin. You can see like a reddish area with a raised ring. So that's why it's called ringworm. And then the little kids are affected the most because they like to play a lot with dogs and cats, and then they rub the fur of these animals, and then they rub their hair, and then they end up with this ringworm. And then we have opportunistic mycosis, which are basically what it causes systemic mycosis. It happens frequently in persons who have AIDS and other people with immunosuppressive disorders. And these will be type of fungal infections that are harmless unless if the person has normal immune system, unless the person has immunosuppression. Example, pneumocystis. Infection in the lung is caused by pneumocystis jirovecii. Okay, so here we have kind of, I guess, an overview of some pathogenic fungi for Mucormycota and Microsporidia. They can be systemic in the case of Mucormycota, or in the case of Microsporidia, it's more localized, but still is serious, like diarrhea and keratoconjunctivitis. In the case of microsporidia, it can cause, for instance, death in someone who has AIDS because the person has like kind of immunosuppression. It can die of diarrhea really easily, as happens with some protozoans like Cryptosporidium parvum. And then for Ascomycota, we have Aspergillus and Claviceps purpurea. So habitat for Aspergillus is found everywhere. That's why it's called ubiquitous. And Claviceps, it is found in grasses. So Aspergillus can cause systemic infections, and then Claviceps purpurea causes toxins that can also lead to serious medical condition in the person. Aspergillus flavum. I was trying to remember the species. So this one causes a toxin that causes liver cancer. And grows in rice, Aspergillus flavum. Ascomycota, we have several species. We have Blastomyces dermatiditis, Histoplasma capsulatum, Stachybotrys, Sporotris chenki, Coccidioides simitis. Most of them are found in soil, as you can see there. And in the case of Histoplasma and Blastomyces can be also found in decaying matter. All of them, with the exception of Sporotris chenki, causes systemic infections. Sporotris chenki, this type of fungus, it is found sometimes in rose bushes, and when gardeners are cutting roses or taking care of the rose bush, sometimes they stab themselves a little bit with a thorn, and then they end up with a lot of inflammation that doesn't go away. Well, sometimes these rose bushes can be contaminated with Sporotris chenki, and then the person has sporotrichosis. And then for Ascomycota, we have Microsporum, we have Trichophytum, and Epidermophytum. All of them are dermatophytes. They live in soil, and also in this case for Microsporum species and Trichophytum, they are found in animals. So these are zoonotic diseases as well. So they can cause like the ringworm. And then Epidermophytum, it's only found in humans and causes all superficial infection, cutaneous infections. And athlete's foot is caused by this species, Epidermophytum, Trichophytum. Then we have one specific that is called Microsporum canis, that it is found in dogs, that's why it's called canis. Trichophytum, I don't remember the Mentagrophytis. This is another one that causes ringworm. And then why, if you take a shower in hotels and all that, it's advisable, I know it's ridiculous, but it's advisable to wear sandals. Because, I mean, you will end up with athlete's foot. And then, I don't know who does that, but some people do, not sharing underwear. They do, and they end up with some mycosis in this area, in the inguinal area, which by itself, guys, it is recommendable not to use so much tight clothing, and then because it can lead to underwear, because it can lead to lack of release of the body temperature, and that leads to formation of moisture that it is used by this species. Candida albicans is Ascomycota. It is found in humans. We have new species that has been found, like auris, also infect humans. And then we have pneumocystis jirovecii, that it used to be called carini. This one causes pneumonia. This is an opportunistic infection. Same for these two. And then when it comes to Candida albicans, causes basically infections in the skin, like diaper rash. It is caused by Candida albicans. In females, can cause vaginitis, oral thrush, and it is found very frequently in immunosuppressed people. And then for Candida auris, it is one of these fungi that it was found like 2 to 3 years ago. It is highly resistant to antibiotics, so or antifungals, so they have been doing some research in it. Cryptococcus. Cryptococcus, we have these neoformans that can live in soil, beer feces, and causes systemic infections, like lung infections. It can go also into your brain. Malassezia is another Basidiomycota. We have Malassezia furfur, something like that. I don't remember. I think so. It's not purple. It's furfur. Malassezia furfur. This causes a skin infection that is called tinea versicolor. Actually, most of the skin infections are called tinnias. And then this tinea versicolor affects the trunk area, and it gives kind of different colors. Like sometimes you have tones of skin that look yellowish, tones that are purple, bluish. That's why it's called versicolor, because it has many colors. And then also can infect the scalp. And then Amanita. I already told you about this, Amanita phalloides versus Muscaria. Muscaria is toxic, phalloides is not. To know the difference, I mean, it's a better idea just to try eating all of these things. So, economic effects of fungi. Well, Aspergillus niger, although it can cause allergies, as any other Aspergillus, I guess, any other fungi species, causes allergies, respiratory allergies in persons. Aspergillus niger is actually used for producing citric acid, which is used in the food industry to preserve foods. Aspergillus terris, it is used to produce statins, which are drugs that lowers cholesterol. Saccharomyces cerevisiae, it is used to produce bread, wine, and also its genome, it has been used to produce hepatitis B vaccine. Trichoderma has this enzyme, trichodermal species, cellulase, that can make our garments or the fabric for the garments that we use to make it softer, because it can break down cellulose. And then we have Taxomyces that produces a drug that is called Taxol, and it is medically speaking important. We have Toliplocladium inflatum that produces this drug cyclosporin. And then we have Conidiotherium minitans that kills fungi on crops. And then Paecilomyces that kills termites. Here is a view microscopic scanning electron microscope of Candida albicans. So as you can see, these are parent cells that they have bud a lot. Now for lichens. So lichens are specific forms of algae that are combined with fungus It's green algae combined with fungus. Now, or it can be a cyanobacterium or in fungus. So we have for lichens that lives in the environment three types of shapes. We have some that are called crustaceae that are encrusted on a substrate. And then we have some that looks like leaves. These are folios. And then some that looks like finger that are called the fructicose. So these lichens, they have a body that is called the thallus, and within the body you have medulla, which is hyphae grown around the algae cells. So you have the algae and then you have your hyphae around, and then you have something that is called the rhizines or holdfasts, which are projections of the hyphae that is located below the body. And then in the cortex, which is like the outer part, you have a coating over the algal layer. So here are the three types of lichens. So this is a branch of a tree. So this is the one that is attached to the substrate, which is the crustose. And then you have here the folios, and then here you have the fructicose. So if you remember the shape, so folios looks like a leaf, and then this looks like a finger. If you see internally, in the lichen thallus, in the medulla, you have the symbiosis. So here, in this case, you have an algae. It can be also a cyanobacterium, that it is embraced by this fungal-like hyphae. And then here you have the cortex surrounding your medulla. And then if you extend this microscopic view of the cortex, you can see in this area how some of these fungus connects into the cortex, and then you have the rhizine emerging from the cortex surrounding all of this, the body. So the algae produces carbohydrates in these lichens, while the fungus provides the holdfast. Economic importance of these lichens is that it helps to form dyes. It has sometimes, some of them, antimicrobial properties. For instance, we have this usnic acid that is formed from a species of lichen that is called usnea. And then litmus. Litmus is a chemical that is used to do this litmus test. And lichens are used by herbivores to survive. Any questions? Algae, the other type of eukaryotes, they don't have a taxonomic group. I'm pretty sure that there is not so much variation. That's why they don't make this taxonomy classification. They are either unicellular or they form filaments. And these filaments are photoautotrophs. So they use CO2 and light to produce energy. They don't have roots, stems, and leaves. Most of them, they live in aquatic environments, and when they don't live in aquatic environments, they typically form symbiosis with the fungi to form these lichens, and water is necessary for growth and reproduction of algae. So we have subclassifications like brown algae. We have diatoms, dinoflagellates, water molds, red algae, and green algae. When it comes to red algae, this one, it is important because it produces toxins. there is one type of algae that can live in symbiosis with some puffish, and some people where this puffish is located, they like to eat that, but there is a warning in these areas, in the beaches where this can be found, that they put a seasonality warning, like, okay, red tide warning, don't catch puffish. Because unfortunately, some people have eaten this puffish, which basically eats the algae and then it releases toxins. And then when the person eats this fish that was catch in the wrong season, then they can get the toxin going into the bloodstream and then they can die. Now, green algae, it is not poisonous for us, and it has an importance for us because this one has been genetically modified to produce biofuels, like ethanol, for instance. So location of the algae will depend on where the nutrients are available, wavelengths of light, and surfaces to attach. Now, the vegetative structures of algae will be a thallus, which is almost similar to the thallus of lichens. It has holdfast, stipes, and blades. And then the cells that cover the thallus will be the ones that are in charge of photosynthesis, and then they absorb the nutrients or water over the entire surface. And then they can have some internal structures that are called the pneumocysts that helps, like the cylinders that some bacteria have, like some cyanobacteria, to go up and down along this aquatic surface to catch sunlight as they need it. So here we have, according to the depth in the ocean, habitats for different algae. So the surface will be 0 meters up to 300 meters. So here within this area, 250 meters depth from the surface of the water, we have penetration of light, and then we have blue blue light going here in this area, and then the red algae has a specific chlorophyll that allows them to live within this area. And then we have violet light here that also it is used by the more superficial red algae. And then here where we have red light, it is used by brown algae, and then green algae lives more toward the surface. And we have, of course, in the land, green algae, and then we have cyanobacteria and euglenoids, which is protozoans, of course. Life cycle. So all can reproduce asexually, all algae, and then we have some algae that are multicellular that can fragment, and then they can reproduce sexually. So they can alternate this reproduction. And then nutrition, most of them are photosynthetic, and they have chlorophyll A that helps them to catch the rays of light, and then they produce this photosynthesis. And then with this type of chlorophyll, they can form different colors of the algae. And then you have some algae that are chemoheterotrophic. In this case, you call it oomycotes. So here we have these unicellular green algae, or Chlamydomonas, that can either reproduce asexually or sexually. So when it reproduces asexually, it does so just by mitosis, and then they grow and they form this large form with this flagella. And then internally, you have the nucleus, and then you have this chloroplast and starch that is stored in the cytoplasm, and then you have something that's called the eye spot. So this one can form gametes to reproduce sexually, and then the gametes, they fuse, they fertilize, they form a zygote, and then the zygote undergo meiosis, so you form more gametes, and then you grow more algae. Brown algae, another name is scalp. It has cellulose and alginic acid in the cell wall. It is multicellular and macroscopic. It can reach lengths of 50 meters, so they can be very long. And then they produce algae, which is a thickener in foods. So this is where agar comes from, guys. And then again, you can watch that video from ASM, American Society of Microbiology, from that webpage that I put there, the link in Canvas. So this is a view, microscopic view of microcystis. This is a brown algae. So this will be the stipe, which is like similar to the filaments that we have in hyphae. It will be the long extension. And then out of these stipes, you have the blades. That looks like the extensions of the feathers. And inside these blades, next to these stipes, you have pneumacyst, which is basically a cylinder that is filled with air that helps for this to move up or down to catch the rays of light within the ocean or the lakes where they live. Red algae has branched thalli. Mostly is multicellular, and it has the ability to live in very deep in the ocean or in lakes, and is harvested for agar as well, and for also to grab a pigment that is called carrageenan. And some, they produce little toxins. I mean, it's not that all species produces these toxins, but that pigment, carrageenan, goes into the pupfish when it eats them. And for the pupfish, it's not toxic. Otherwise, we wouldn't have pupfish. But for humans, it is. And then the toxin, of course, not the pigment. But that's one way to tell. And then here we have microcladia, which is a type of algae. So this has branched thalli, so this will be the thalli. Looks very beautiful. It looks like a tree. Green algae has cellulose cell walls, can be unicellular or multicellular, has both types of chlorophylls, A and B, store starch, and give rise to terrestrial plants. Has two types of reproduction as well, sexual and asexual. So undergo mitosis, grow and form this mature form. And this mature form can form gametes. The gametes will fuse. They form the zygote. This zygote undergo meiosis, form more gametes, and then they form the adult form. And here we have a multicellular green algae, which is called ulva. Euglenoids are photoautotrophs. And they have an eye spot and a flagellum that it is used to seek out light. And then diatoms, we have cell walls with pectin and silica, and they are unicellular or filamentous. And then they store oils in their cytoplasm, and they produce an acid that is called domoic acid that can cause problems in the brain, neurological disease. Here we have Eunotia, which is a freshwater diatom. So it has this rectangular shape with a ring in the middle, or well, a stripe in the middle, and it has like these little pores. So this type of diatom grows in acidic water, and it has a sexual reproduction, combining with another structure. Dinoflagellates has cellulose in the plasma membrane. They are unicellular organisms, and they're part of the plankton. So they are these also flagellates that can be ingested by pupfish, and they produce this neurotoxin that is called sagittauxins. So these cause paralytic shellfish poisoning. And again, this is one of the ones that it is very important for people to not ingest this type of shellfish when they are contaminated with these dinoflagellates. Here we have perineum. So it has like this rhomboid shape. In the middle has this depth structure that is called the flagellum that exits as well through this middle piece. That's the bottom view. All Mycota are water molds. They have cellulose in their cell walls, and they are chemoheterotroph. They reproduce by the release of zoospores, and they are closely related to the diatoms and dinoflagellates and to fungi. They decompose decay matter and are plant parasites. So we have Phytophthora infestans. This one, when was it? 1600s or 1700s. Destroy the crops of potatoes in Ireland. And then basically potatoes, it's one of these very important foods in the diet of Irish people, and many of them, they die because they didn't have anything to eat. And then we have Phytophthora cinnamoni, which infects plants like eucalyptus. And then we have one that infects oak, which is Phytophthora ramamorum. Okay, we will leave it there.